Imagine a petri dish in a lab. Inside this dish, there is no sperm. There's no egg. There's literally no womb.
Just a carefully calibrated liquid.
Right. Yet floating in that liquid, a microscopic cluster of cells is spontaneously organizing itself. It's folding into a tiny, rudimentary neural tube. And then, incredibly, a microscopic, heart-like structure actually begins to rhythmically beat.
I mean, it sounds completely like science fiction.
It really does. But this is happening right now in a lab. Welcome to today's deep dive. If you're listening, go ahead and grab a coffee because our mission today is to unpack a truly groundbreaking 2026 paper.
Yeah, published in the journal Cell by Zhang and colleagues. It's titled Continuous Modeling of Primate Embryogenesis from Totipotency to Early Organogenesis.
Right, and we are going to break down every single mechanism and term step by step. Because if you've ever wondered how a loose cluster of cells inherently knows how to build a living body well, you are about to find out.
It's a question that has baffled developmental biologists for decades, Early embryonic development, especially in primates, has historically been a complete black box.
Because it happens so early and so deep inside the body, right?
Exactly. We're talking about events that occur just days after fertilization, and the structures are smaller than the head of a pin. But this paper gives us a window into that exact process. By creating a fertilization-independent system, we can actually watch the architectural blueprints of life unfold in real time.
Outside of normal reproductive constraints, it's wild. Okay, to understand how a loose pile of cells turns into a beating heart, we have to start at cell zero. Usually we hear the term pluripotent stem cells, but this research specifically focuses on totipotency. What is the fundamental difference there?
Well, it really comes down to developmental potential. Pluripotent stem cells are incredibly versatile. They can differentiate into pretty much any cell type that makes up the actual adult body, muscle, bone, neurons, you name it.
But they have limits.
Right. Totipotent cells possess one additional critical capability. They can create all the cells of the body, plus the extra embryonic tissues necessary for that body to survive in the womb.
Meaning the placenta and the yolk sac.
Exactly.
Okay, I like to use a mnemonic for this. I've always thought of pluripotency like having a house key. You can get into the kitchen, the bedroom, the living room. You have access to build any room inside the main house, which is the embryo body.
I like that analogy, yeah.
But totipotency is the master key. It opens the house, but it also opens the front gate and builds the guest house. It builds the embryo, but also the placenta and yolk sac. Without that master key, the house can't actually function on the property.
The master key analogy works perfectly. But with one crucial caveat, these cells don't just unlock doors, they actually possess the inherent instructions to build the doors and the walls as they go.
Oh, wow. Okay.
Yeah, and in natural development, true totipotency only exists very briefly, like in the zygote right after fertilization and maybe the 2 to 4 cell stage. After that, the cells divide, specialize, and effectively trade in their master keys for house keys.
So what Zhang and the team did was artificially create and stabilize cells holding those master keys in the lab.
Right. They call them cTBLCs, which stands for cynomolgus monkey totipotent blastomere-like cells.
Cynomolgus monkeys being the standard primate model that closely mirrors human biology. But here's the thing I'm trying to wrap my head around. They didn't just harvest these master key cells from actual monkey embryos, right?
Oh, no, they didn't. They started with standard primed pluripotent stem cells.
The ones that only have the house key.
Exactly. And they somehow reverse engineered them back into master key cells.
How do you force a cell backwards in time like that?
It requires a highly specialized chemical bath. They called it LMYCIP culture medium. The name itself is just an acronym for a specific cocktail of inhibitors and growth factors.
Okay, so a chemical soup.
Right, they use chemicals to block various cellular pathways.
Oh.
But rather than getting bogged down in the whole alphabet soup, the absolute secret sauce, the thing that forced the regression, is a molecule called platianolide B.
Platianolide B. What makes that one specific molecule powerful enough to hit reverse on a cell's development?
So it acts as a splicing inhibitor. To visualize why that matters, think about how a cell reads its DNA. When a cell reads a gene, it creates a rough draft of messenger RNA.
Okay, like a rough blueprint.
Right, but that rough draft has a lot of extra non-coding genetic junk in it. It has to be edited or spliced to remove the junk before the cell can use it as a final blueprint to build proteins.
And there's machinery in the cell that does this.
Yes, the biological machinery that does this editing is called the spliceosome. What platianolide B does is it essentially jams the gears of the spliceosome. It causes widespread spliceosome repression.
So you're physically stopping the cell from editing its own genetic blueprints.
Exactly.
But hold on. If you violently chemically paused a cell's RNA splicing machinery to force it backwards, hasn't that damaged the cell? How do we know it isn't just biologically confused? I mean, how do we know it can actually function as a master key and not just like a broken lock?
That skepticism is exactly what the scientific community demands. You can look at the RNA readouts and say, well, it looks totipotent on paper because the specialized genes are silenced. But that's just circumstantial evidence.
You need proof.
Right, you need functional proof that it can build both the house and the gate. So to prove it, the researchers performed the gold standard test, the chimeric mouse assay.
Meaning they injected these monkey cells into a mouse.
Yes, they took these lab-created monkey cells and injected them into a living 8-cell mouse embryo.
Wow.
If the monkey cells were only pluripotent, they would only contribute to the mouse fetus itself. But they tracked the monkey cells using fluorescent markers, and they found them turning into trophoblasts.
Which is the scientific term for the precursor cells that eventually build the placenta, right?
Exactly.
Yeah.
Trophoblasts build the placenta, and they also form cells that build the yolk sac.
So they actually integrated and formed the life support structures. That's incredible.
It is. And they went one step further. When injected into immunodeficient mice, these monkey cells formed teratomas.
A teratoma is a type of tumor, right?
Yeah, it's a tumor that contains fully specialized tissues from all 3 fundamental germ layers. Things like cartilage, gut lining, and neural tissue, all just growing chaotically together.
That sounds bizarre.
It does, but in biology, forming a teratoma is the ultimate proof of maximum developmental potential. Functionally, these cells were the real deal.
Okay, let me pause you there to make sure I'm tracking the biological logic before we move on. Time for a quick recap and bullet summary for you listening at home. Bullet 1, pluripotent, means embryo only, while totipotent means embryo plus placenta. The house key versus the master key.
Spot on.
Bullet 2, cTBLCs are lab-made, totipotent monkey cells created via splicing repression using that palladium B in the LMYCIP medium. And bullet 3, they proved their master key status by successfully growing into both embryo and placenta tissues in living mice.
That's a perfect summary.
Awesome. So, transition time. We have the raw materials. We have these powerful master key cells. What happens if we just wash off the chemicals, put them in a dish, and leave them alone without any instructions?
That is where the biology gets truly beautiful. In standard stem cell biology, if you want a cell to become something specific, you have to hit it with a heavy dose of external signaling manipulators to force its hand.
Like pushing it down a specific path.
Right. But here, when the researchers placed the cells in a basic culture medium called IVC1, they didn't need to force anything. Over about 8 days, the cells underwent spontaneous morphogenesis.
Meaning they built their own structure.
Yes, they self-assembled into blastoids, which are highly organized hollow spheres that look and act exactly like natural early embryos.
But structurally, how? How does a cell in a loose pile inherently know to form a tight hollow ball?
It relies on 2 mechanical processes, compaction and polarization. Let's look at compaction first. The cells begin to express a protein called E-cadherin.
Okay, E-cadherin.
Think of E-cadherin as biological Velcro. Initially, this Velcro is spread loosely all over the cell surface, but around day 3, it concentrates specifically at the contact points between the cells.
Oh, I see.
Yeah, the proteins bind together, physically pulling the cells tightly into a solid, compacted mass.
Like pulling the drawstrings tight on a bag. That makes sense for clumping them together. But a blastoid isn't just a solid clump, is it? It's a sphere with an outer layer and an inner cluster.
Yeah.
So how do the cells figure out who goes where?
That is polarization, and it's entirely driven by physical geometry. A protein called YAP is the key decision maker here. Right. Around day 4, the cells have to figure out if they are trapped on the inside of the clump or if they are sitting on the outside edge. The cells on the outside experience a totally different physical tension. They have a free surface facing the liquid, whereas the inside cells are squeezed from all sides.
So the physical squeezing literally changes the cell's behavior.
Exactly. This physical tension causes the YAP protein to physically relocate into the nuclei of only the outer layer cells.
And once YAP gets into the nucleus, it acts like a genetic switch.
You got it. It enters the nucleus and turns on a specific genetic program that says, hey, you are the outside layer. You'll become the trophectoderm, the future placenta.
And the ones on the inside.
The cells trapped on the inside, which don't get YAP in their nucleus, remain the inner cell mass, which will become the body. That is the very first cellular fate decision. and it happens entirely spontaneously based on physical positioning.
Wait, here's where it gets really interesting for me. Because earlier you mentioned that in natural embryos, there is a massive genetic event right after fertilization, a sort of factory reset of the cell's hard drive to kick off development. Did these lab cells do that?
You're thinking of zygotic genome activation, or ZGA.
Right, ZGA.
In natural biology, when a sperm meets an egg, the egg is packed with the mother's RNA. Her instructions to help the cell survive the first few days. ZGA is the biological process that wipes out the mother's RNA and boots up the unique, fresh genome of the newly formed embryo.
It's the starting gun for life.
Historically, yes, it's considered the absolute initiation point of embryonic development. We fully expected these lab-grown blastoids to go through that exact same factory reset using standard factors like ZSCAN4.
But single-cell RNA sequencing showed they didn't.
They completely bypassed it. As these cells were self-assembling, they passed through a transitional phase called the 8-CMLC stage. That stands for 8-cell marula-like cells. Crucially, these 8-CMLCs completely ignored the traditional ZGA factory reset genes.
So if they skip the main power switch that natural embryos use, how do they boot up at all?
They rely on a totally different built-in genetic network. It's heavily driven by genes like ATF4, DDIT3, and crucially, a gene called BTG1. Think of BTG1 as an emergency backup generator that boots up the house when the main power, the fertilization reset, just isn't available.
Because they never had a mother's RNA to begin with.
Exactly. The researchers realized that because these lab-made cells never went through a sperm meets egg fertilization event, they didn't have any maternal RNA to clear out. They simply didn't need the traditional ZGA factory reset. They possessed this alternative latent program to initiate life.
But a hypothesis is one thing. How do we know this alternative BTG1 emergency generator is actually what's driving the bus and not just some random background genetic noise?
Through an elegant experiment called a knockdown. Using siRNA, which is small interfering RNA, the researchers essentially created a genetic volume knob. They turned the expression of the BTG1 gene down to 0.
And what happened?
When they knocked down BTG1, development derailed disastrously. The cells couldn't form the proper inside and outside layers, and bizarrely, they started activating muscle genes way too early. They completely lost their architectural map.
And just to be completely rigorous, did they also try turning down the traditional ZGA factory reset gene, like maybe knock down NR5A2?
They did exactly that. They knocked down the traditional ZGA gene, expecting it might cause a problem, and nothing happened.
Nothing.
Nothing. The blastoids formed perfectly. This proves beyond a doubt that there's an intrinsic developmental initiation program coded into these cells. They possess a sequential regulatory network that can build an embryo completely independent of the classical mechanical requirements of fertilization.
Okay, mind blown. Let me summarize this second segment before we get to the really crazy organ stuff.
Yeah.
Here is your second bullet summary. Bullet 1, cTBLCs spontaneously self-assemble into blastoids through the mechanical processes of compaction and polarization. Bullet 2, they completely bypass traditional fertilization factory resets, meaning they skip normal ZGA. And bullet 3, instead, they rely on a unique intrinsic network, specifically the 8-CMLC stage, driven by backup generator genes like BTG1 to decide their fates.
That is a flawless summary. So now we have a self-assembling blastoid, which is essentially a highly organized hollow ball of cells. But a hollow ball is not a body.
Right.
To get to the beating heart, the embryo must undergo one of the most dramatic physical transformations in all of biology.
Gastrulation.
Yes.
This is where things go from a 2D sheet to a 3D organism. The embryo organizes into the 3 primary germ layers that will eventually form every tissue in the body. The ectoderm on the outside, the mesoderm in the middle, and the endoderm on the inside. How does a simple sphere of cells actually achieve that?
It starts with the formation of something called the primitive streak. It is essentially a groove that opens up on the surface of the embryo where cells from the surface begin to migrate inward.
Okay, I picture gastrulation less like a groove and more like a cosmic sinkhole, or like an incredibly complex origami folding process. The 2D sheet of cells physically buckles inward under its own tension. It tucks specific cells deep inside to become the endoderm, which forms the gut.
Yeah, that's a great way to visualize it.
And then other cells dive into the middle to become the mesoderm, forming muscles and blood. And the cells left safely on the outside surface become the ectoderm, which turns into the skin and the brain.
That origami analogy is spot on because it highlights the physical, mechanical folding of the tissue. But to get our lab-grown blastoids to perform this origami, the basic culture medium wasn't enough anymore. The metabolic demand of building organs is massive.
So they needed more fuel.
Right. So around day 15, the researchers switched the culture medium. They added rat serum and a precise glucose gradient to act as that nutrient fuel.
And the results they got between day 20 and 23 are the reason we are doing this deep dive. Let's walk through the actual structures that formed in that dish, starting with the brain.
Well, the very beginnings of the brain. The neural ectoderm, those cells from the surface layer of your origami fold, began to physically fold inward to form a distinct neural tube.
Just spontaneously.
Spontaneously. It perfectly mimicked natural neurulation, showing a closed posterior end and an open anterior end. And when they analyzed these cells, they found them expressing PX6.
What is PX6?
It's essentially a molecular name tag that identifies a cell as a neural progenitor. You are literally watching the foundational architecture of the brain and spinal cord assemble itself.
From the gut, because you need a gut.
The endodermal cells, the ones tucked deepest inside, didn't just form a random generic tube, they regionalized. They organized along an anterior-posterior axis, creating a distinct foregut and hindgut. Again, they confirm this by looking at specific molecular name tags, like FOXA2 and CDX2, which mark gut development.
And then the absolute climax of the entire experiment, the heart structure.
This is where the model truly pushes the boundaries of what we thought was possible in vitro. A specific group of mesoderm cells began to specialize into cardiogenic tissue, marked by genes like NKX25 and GATA4.
Heart genes?
Yes, they actively proliferated into a multi-layered myocardial mantle. They formed a primitive endocardial tube. And because these cells were functionally maturing, this tube exhibited spontaneous, rhythmic, cardiac-like beating activity.
Right there in the dish?
Right there in the dish. Furthermore, while this heart tube was beating, they also observed the dynamic migration of primordial germ cells traveling along the surrounding tissues.
Wait, primordial germ cells, meaning the precursors to sperm and eggs, the cells that create the next generation.
Exactly.
Okay, I have to play devil's advocate here. If we have a beating heart, a folding neural tube, a regionalized gut, and the literal cells required for reproduction, are we basically growing a monkey in a lab dish? Because that sounds like artificial womb territory.
It is a totally natural question, but it's crucial we clarify the strict biological limits of what is happening here. No, we are not growing a monkey in a dish. This is not a fully formed fetus.
Okay, explain the limits.
What Xing and the team created is a highly advanced model that reaches the stage of early organogenesis. In human developmental terms, that's roughly comparable to Carnegie stages 8 to 11.
So they hit a biological wall. They don't just keep growing indefinitely.
Exactly. When the researchers performed single-cell sequencing on these late-stage embryoids, they found that certain critical cellular populations were just missing.
Like what?
Well, for instance, parts of the paraxial mesoderm that are required to form the somites in the spine were incomplete or totally absent. The model is incredibly accurate up to a specific point. But eventually, without the massively complex dynamic maternal environment of a real womb, the structural integrity breaks down and development halts. Got it. Okay, so to pull together this final stage, here is your third and final bullet summary. Bullet 1. By changing the environment, giving them rat serum and a glucose gradient, the blastoids enter gastrulation, folding themselves into the 3 essential germ layers.
Right.
Bullet 2, they successfully form early organ structures, a folding neural tube, a regionalized gut, and migrating germ cells.
Yeah.
And bullet 3, amazingly, they develop a beating cardiac tube, though development naturally hits a wall and halts shortly after this stage.
Which brings us to the ultimate question for you, the listener. If this model hits a wall and stops growing, why is this 2026 Cell paper so revolutionary for you?
Why does it matter?
You should care, because early embryonic development has historically been a tragic mystery. Miscarriages most frequently occur at these exact hidden stages. The foundational errors that lead to severe birth defects originate right here, during compaction, polarization, or primitive streak formation. And because natural embryos are microscopic and we have obvious ethical and physical barriers to studying them inside a human body, we've basically been trying to solve these medical mysteries completely in the dark.
Precisely. But now, thanks to these totipotent master key cells, scientists have access to an unlimited, highly accurate, and directly observable model. We can use this to study the genetic origins of developmental disease. We can test the safety of pharmaceutical drugs on early tissue formation without risking human life.
It's unbelievable.
We can fundamentally understand the mechanics of human reproduction in ways that were practically impossible even 5 years ago.
It really is a completely new frontier for medicine, and I want to leave you with a philosophical and biological mystery to ponder long after this deep dive ends. If you take a single cell, bypass the sperm and the egg entirely, remove it from the body, and yet it inherently possesses the architectural blueprints to fold a neural tube and rhythmically beat like a heart, what other latent ancestral programs are hiding inside our cells, just waiting for the exact right chemical environment to wake them up?
It profoundly changes how we view the memory and capability of the individual cell, doesn't it? We used to think the cells is just a brick in the wall. Now we know the brick contains the blueprint for the entire city.
So true. So if you could look inside one of these lab-grown models, peering through the microscope, which organ system's origin story would you want to watch unfold first? The folding brain, the beating heart, the gut? Let us know. And next time you look at a seemingly simple cell, remember that petri dish and the blueprints hiding inside?